Morning. Really good to be here again. I'm going to speak today about ways to look at the signals that we have from a variety of techniques, what we call omics techniques, that are exploring the gut immune brain axis and really trying to see how we can use these and why we might want to use these signals to try to understand how the heterogeneity, all this diversity among individuals with ME/CFS may relate to what we can learn about the pathways to disease. So why phenotype? Why do we go there? I mean, we really don't know for certain whether if you were going to say that ME/CFS is ice cream, okay, and then there may be all of that variation, maybe just different flavors of ice cream, and maybe comorbidities that people have are just sort of whether you're putting a topping on. But the fundamental issue would still be ice cream. And so the question is really what pertinence does that diversity have to understanding what causes the disease, how you might prevent the disease, and also what the prognosis is and how you might treat it. So one of the ways to look at this is really to try to really observe as closely as we can and to gather as much information as we can on clinical aspects of the disorder, not forgetting to ask about all the different features, even if you're not certain that it's involved. This is a multisystemic illness, we know that. Perhaps we really do need to be very thorough in what we're asking about. And so these are what we sort of call endophenotypes. These are really thought to relate to sort of a gene environment. It could be a genetic factor or a gene environment interaction that is one pathway to disease. So here you have all of these different these three separate endophenotypes. There may be some variety even within each of the endophenotypes. And as an example of the way one might think about this, you could think about ME/CFS with or without POTS, with or without fibromyalgia, with or without Ehlers-Danlos syndrome, or symptoms of it. That's come up lately, and some questions about certain types of novel treatments for what at least looks like ME/CFS, and IBS, which is irritable bowel syndrome, or other types of gastrointestinal symptoms. And there's lots of other types of examples of that. And then we can take all these data we're doing, in both from biological specimens, imaging, all the various types of techniques that we have, really getting thorough histories, and then really doing in this pan-omics way and looking at all the exposures and using various machine learning or artificial intelligence, like topological data analysis, which I'll show you some examples of that a little bit later, and then trying to parse these individuals out with these different superficial features to begin with, doing all of these analyses, microbiome, metabolomics, proteins, RNA, and genetic factors, epigenetic factors, and then really trying to see how these clusters, which are just, suggestions, if you will, hypotheses as to what might be pertinent, to really think about, okay, for this particular cluster, would you want to change the health strategies that you use to keep the person under, the best conditions and get them to the best place? Are there certain biomarkers that vary across the clusters or some that overlap? Prediction, prognosis. Also, if we want to try to examine who responds to what types of medications, perhaps you want to enrich certain subsets or at least people within these different clusters in order to gather that information for the future, and so forth. And also really tailoring environmental exposure is a little kind of peculiar usage of the word, but it shows really that there can be certain environmental factors that may be good for certain people and many that are probably bad. Mold exposure, for example, for some. Even certain aspects of psychosocial stressors, or managing one's energy levels or activities in order to optimize the maintenance of the best energy levels that one can have. One of the subsets that we've become very interested in is gastrointestinal comorbidity. And this is really an area that, you can't go to any even standard supermarket, at least in the States, without seeing the section with hundreds of probiotics. In the States, you go to Whole Foods, it's an organic food store, and there'll be a wall of like 1,000 different brands. What science there is to tell you which one is right for which individual or whether it actually works for anyone is very sketchy. That doesn't mean that there isn't some important information out there, and I think we have more and more evidence of that. But we also know that gastrointestinal issues are very prominent in all sorts of brain disorders. So it's not just ME/CFS, and so we want to try to understand how does this fit with the prominent, post-exertional malaise, and, or is it really just happen to be there because there's a high comorbidity with irritable bowel syndrome, which can be up to 80% by some estimates. So what do we know, so far? What clues do we have about the why this brain immune or brain metabolomic gut dialogue may be important to understanding this disorder? There's really wonderful, multi-directional effects in these systems that I think do provide strong clues, even though we may not have our probiotics well designed yet to be able to manipulate the system. But what do we know? So, there are stress response system, what we call our HPA axis, which is the hypothalamic, pituitary, adrenal axis, that makes stress hormones, including cortisol being released from the adrenal glands. That's really part of your main response. If you don't have a good cortisol response, we always think of a stress hormone as being bad. If you don't have a little bit of that happening, your immune cells are not going to be able to do their work when you get an infection. Right? Cortisol goes up, and that's really part of it. It actually helps to call out all of the action that you need to contain an infection, but the next step after that is also important, which is negative feedback, dampening things down as well. And we think that there is a good reason to believe that in ME, that it may not be initially at least, the ability to mount a response that is protective, inflammatory response to try to contain things, but it's really turning it down when the danger is no longer there. And we know that there are immune systems that are affected through this, but there's, these microbes, of course, that we live with. You've heard lots about this. They make, using some indigestible carbohydrates that we eat, they make short-chain fatty acids, which, depending on which type it is, can either promote inflammation or promote autoimmunity or protect against autoimmunity, and then are also responsible for creating a whole host of neurotransmitters and other factors that are really important in our brain function. Some of them have transporters, so they can get to the blood through a system that's meant for them to get from the intestine off into the blood and then to the brain. But we can also have leaky gut based upon changes in the little proteins that stand in between these cells in the intestinal epithelium that make up sort of a hole, if you will, so that the various products can get through. So you can have a combination of both. And these, we'll talk a little bit about some of these factors later. There's also immune factors that are made as a result of your interaction of your immune cells as they traffic through the gut. 80% of every immune cell that you have that's in your peripheral circulation makes its way, it does a little traffic route into your intestines every day and picks up messages from the bacteria that are there, and also give the bacteria that are there and the cells that are in the intestines additional messages. So that interplay is very important. Okay. So what else might we know but might make sense about how, at least for the patient who has irritable bowel syndrome type of symptoms along with ME/CFS, what do we know about how the microbes play into this and how the metabolites that they make specifically? Many of the metabolites that we're interested in are ones that only the gut bacteria can make. Humans don't make them. So we can, those have some special, seem to have some special functions. And so there's a sort of a tight organization here. These are involved in GI motility, what we call peristalsis, right? Normal digestion and your ability to get your food from A to B, but also to actually do the process of digestion. And it's also probably important in not only as a result of a healthy autonomic nervous system or vagal nerve system and balance between sympathetic and parasympathetic, but it's also it also goes back again and has different effects and signals back to the autonomic nervous system. So what do we know? So we know that there are certain bacteria that produce methane. And give you gas. That's what those luminal gases are, and that happens in certain places where only certain bacteria may be found at certain levels within the structure of the intestinal wall. And there are different products that are created. LPS is a product that is lipopolysaccharide. It's a product on a bacterial cell that has really important effects at receptors that are called toll-like receptors. Happens to be the toll-like receptor 4 that it's likely to affect, and that will increase GI motility. So if you're not having those bacterial signals, which are inflammatory signals, and they're also active during a bacterial infection too, but it's important in GI motility. Another effect on motility and peristalsis is through serotonin, or 5-HT, and there's something called tryptamine, which is a bacterial metabolite. And also similarly, we talked a moment ago about the short-chain fatty acids that are what bacteria produce after they eat up what is indigestible to the human, these indigestible carbohydrates, and then create these products that also affect the motility. We endeavored about, I guess it's about a year ago, 2 years ago now, to look at the microbiome in subjects within a study called the Chronic Fatigue Initiative, this extension study. And we had 50 patients and 50 controls. And we used an artificial intelligence technique. We sort of put all of the microbiome data, and we get gobs and gobs of it, and we get information even about the gene expression within the bacterium, not only to identify which bacterium it was, but also which metabolomic pathways may be activated within those bacteria. And so each of these circles represents one or more patients. It's the way in this is sort of clusters these data together. And a line between two dots means that there's something shared across those different patient groups. And and those structures form as the different data, seems to associate with itself within your data set. And the gray here are the controls. The ones that are isolated are similar to one another within the dot, but they are not sharing very much with any other subjects. And then the chronic fatigue subjects without irritable bowel are in the red. You sort of see this kite-like structure here, and then some that are solo. And then the subjects without IBS are in the blue. And what we found is that there were really distinct differences even within the ME/CFS group, depending on whether they had irritable bowel syndrome or not. And this is a very long list. It's not intended to actually, memorize this list, but there are some that are, that differ across. Some are shared across the ME/CFS. In purple here is the total ME/CFS group. Red are the ones with IBS, and then this blue are the ones without IBS. And some of them are associated, interestingly, there's some that are higher here that are, that may be an inverse, a backwards marker, if you will, of hypotension, low blood pressure. Marvin-Brantia species are ones that are higher in the subjects, in the cases, and that's something that's usually low in patients that have hypertension. And so, so we know that patients with ME generally have the opposite problem, which is hypotension or low blood pressure or difficulties maintaining it across positional changes. And a variety of other agents are looked at as ones that are considered to be like fecal bacterium. Some of these are thought of as being sort of healthier ones, and those tend to be lower because these are in the gray or the control levels here. We also then took the predictions. We didn't know for sure what the level or the quantity was for each of these different metabolites, but these were the genes that were associated with each of these bacteria to get a sense of which pathways may be likely to be activated among the bacteria within the GI tract, or at least within these stool samples that we analyzed from the total patient population, the patients with IBS and the patients without IBS. And what we found was that there were some pathways like that. Vitamin B6 biosynthesis, very important cofactor that is an assistant, if you will, in many neurotransmitter functions, so for your normal brain signaling. Also important in many other functions, including support of immune functions. Pyrimidine ribonucleoside degradation means that there's a lot of turnover in that. Usually that's at least from what we know about that function in brain, is that it's where there's an effort to try to deal with injury or stress, and to try to keep the plasticity of the brain going. And so you see the vitamin B6 is present in the in the subjects as a whole, and in those without IBS. But it's not, you don't see it in the patients with IBS. What you do see in all three groups is something called atrazine degradation. And so we also need to be thinking, besides infection, besides thinking about the microbiome, the bacteria inside of us, we also, atrazine is the most widely used herbicide in the United States at least, and in, I think in Australia as well. I'm not sure about in Europe. And so it's an endocrine disruptor. So, it works to kill, weeds and, and so forth, but it also has a very prominent effect on hormonal systems, estrogen balance in particular. And so we think that that's interesting and very important to look at. We don't know that it's a cause of dysfunction. Usually that's only been examined in, let's say, pregnant women or very early childhood, but certainly something that is worthy of additional look. And so again, putting this together and focusing on that kite-like structure that you saw with the IBS here in the red, the blue without the IBS. These are subjects who had IBS but had a normal BMI as opposed to those who had a high BMI. And so we saw different patterns in the bacteria, the cytokines, as well as the predicted metabolic pathways, and these associated differently with symptoms. And sort of a summary, if you will, of what we saw before, but just sort of pointing out these are, some of these are allergy-associated cytokines, and that there also are, this is associated with high BMI, and leptin is an adipokine, so it's a cytokine that is from the fat tissues. So what did we see here? Why did we do this? What did we learn? We learned that, as others before us had suggested, many others in recent years, that there's some form of intestinal dysbiosis that is present in ME/CFS as a whole. It also varies according to the comorbidity with irritable bowel syndrome. And it also has, the IBS is also the strongest driver. In other words, it explains the most variation in all of the different microbe microbes that we saw. We know that there are differences in the metabolic pathways that were predicted. Again, we didn't measure the metabolites, but this is what we would expect to see. And we saw a decrease in unsaturated fatty acid biosynthesis, I think really important in thinking about how we're getting to energy metabolism. And we also saw this increase in atrazine degradation, B6 biosynthesis, and pyrimidine ribonucleoside degradation. So correlated with pain, fatigue, reduced motivation, and we think that this type of work obviously is not the end-all, be-all. There can be unique things about certain subject populations, so you want to really look more carefully and more widely, and to really describe patients as thoroughly as possible so you know what types of clinical symptoms may be there, and putting various factors together. So again, there's many pathways that we know about. There's so many more that we still need to know. This is really here just as a reminder to that many of the factors that we're able to measure in metabolomics, ketone bodies, and the amino acids, that these are having an interaction with the immune system and shaping the immune system both within the GI tract, while the immune cells are sort of resident there in the intestinal tissue, and then changing the messages or their set points or their thermostat, if you will, of each of those immune cells as they go back into the circulation. So there was an opportunity to use the plasma from these subjects to do a metabolomic study. We found that there were substances that were altered. The blue is decreased metabolites, red is increased, and we had patterns that varied based upon the random forest and various standard things, and using Metamap, which is what this is showing you here. It's just graphed in Cytoscape. And the patterns were, varied again by ME/CFS, but we also found distinct patterns with patients with IBS that were more prominent, and there were more differences than in patients here who had ME/CFS without IBS. They had more limited abnormalities than in the subjects, the total group as a whole, as well as in the patients with IBS. And so ceramide, is there, but carnitine is one that's decreased, and choline. Both of those have been used experimentally, if you will, to see if there can be some improvement in function, and been found to be low in other studies as well. So how we put all these things together, we know that there's the metabolites that go in the bloodstream. We also know that there can be direct effects through the wiring, the autonomic nervous system. The vagus nerve can go directly up to areas of the brain and have an effect, and then it can affect back down. And we know that there are lots of different species of bacteria that are associated with the production of various neurotransmitters. So we really need to understand more of this. These just wanted to remind me to tell you also that these are also making vitamins and important in that regulation. These patterns interact with our dietary pathways, alter tryptophan metabolism, and which microbes you have can also alter something else that's very important in ME/CFS, at least for a subset. Again, maybe it's a phenotype. One of the clusters is autoimmunity. And so we want to be able to get to the point where we're looking at the autoimmune features within subjects and look to see how that may or may not relate to the microbes that are present, or to the short-chain fatty acid patterns that are present based upon whether the bacteria shift towards butyrate production versus other types of production. So again, I have lots of people. This type of work takes a village. So I'm grateful to many of my collaborators, and of course to our funding from NIAID, NINDS, and Chronic Fatigue Initiative, as well as private donors and the Microbe Discovery Project, as well as our long-standing collaborations with Cimarron Research. Thanks very much.
Both those talks are now open to you guys for questions, so who would like it? 5 minutes?
So we have seen that the content in cytokines is different from exosome and plasma. And I was wondering if is it possible that in previous studies maybe the content of cytokines in exosome gave a contribution to the result because we had several studies, and the one by Columbia University, of course, and but the results are somehow different, inconsistent. So I wonder if maybe exosomes, the content in cytokines, might be.
Yeah, there's lots of different ways that one can extract the plasma to get cytokines or use serums. Some of the studies are serum cytokines that would not, so serum wouldn't have the exosomes in them. And depending on how people extracted their plasma, they might have gotten the cytokines from the exosomes in or might not. So there's lots of sources of variability between those different studies. But there's no question that there's still, even so, there's still clearly a difference between what's packed in the exosomes and what's in the plasma. So,
Okay.
Maureen, can you tell, so within the extracellular vesicles, is there a way to distinguish those that are coming from the brain versus the gut versus other places?
that's a really active area of research right now. A lot of people would love to have an affinity purification for cells from different, for exosomes from different cell types. And right now, there isn't a good way to do that that's, gets a lot of the, a good level of purification, but that's really an active area of research. And that's what I'm hoping we'll be able to do, is we'll be able to sample the brain, for example, by fractionating the exosomes.
One more question.
You talked about mitochondrial membrane potential. I wondered, do you know, can the nervous system and the state of the nervous system influence mitochondrial membrane potential and therefore mitochondrial function? And have you looked at that? Has anybody looked at that?
We only looked at mitochondrial membrane potential in the cells that are easy to get, which is the cells in the blood. I don't know of anyone who has been able to, for example, get a brain biopsy and find out whether anything is different there. However, there is brain imaging going on, which can have an indirect assay of the mitochondrial function in the brain by having specific isotopes that bind to a mitochondrial protein. And so we can get some information from brain imaging and mitochondrial function in the brain through for example, PET scans. So that's ongoing research in several labs, including one of the ones that Vicki mentioned is just been funded.
So you don't know if any, say if you change the charge that's on the mitochondrial membrane, do you change the function? And therefore could nerves impact the function?
Well, certainly the mitochondrial membrane potential is absolutely essential for functioning mitochondria. If you don't have any membrane potential, your mitochondrion is non-functional. That's what it definitely needs, the membrane potential. But I don't know of a good way to increase it. There are a lot of bad chemicals that can get rid of your membrane potential, but I don't know of a drug that is extremely helpful for increasing the membrane potential.